Good day. In this lecture, we will be talking about theories of inheritance. So our reference books for the course as a whole is from Hartwell's Genetics, from Genes to Genomes. This is from McGraw-Hill, and also Essentials of Genetics by Klug from Pearson. Okay, so let's first define genetics. I believe you have encountered this one during your high school general biology days. So simply put, genetics is widely regarded as the science of heredity. So it seeks to pursue an explanation of the mechanisms that determine our inheritance. So as you can see here, when we talk about genetics, you can see here inheritance, genes. Actually, genetics comes from the word genes. So of course, you have DNA, sequences, organism, inheritance, heredity, etc., etc. So it can be a simple idea as following a single gene and looking at changes to those genes as time passes, or it can be as complex as the interaction of many different genes. So here is the timeline of the field of genetics that started with Mendel in 1860. Although humans have been tinkering around with inheritance, heredity as way back as ancient times, the formal study or recognition of this one as a science, they was regarded to originate from Mendel's work. That is in 1860s during the publication, but it wasn't until the 1900s that Mendel's work from the early 19th century was actually recognized. So it took about 40 years for that. And you have this chromosomal theory of inheritance that starts to be proposed during that, because during that time major scientific discoveries have started to emerge. Basically, they have discovered the DNA, they have discovered proteins, they are trying to sequence or determine what proteins are made up of. And then in the 1950s and the 1960s, this is the molecular genetics era. So this is around the time where the DNA structure was discovered by Watson and Crick, and therefore it unlocks the different mechanism on how DNA, basically our genes, can be passed on through the generations. And then 1970s, 1980s, you have the recombinant DNA technology. We have started tinkering with our genes, and right now in the 2010s, this is the epigenetics era after the gene. So we are now trying to edit the genome. We are now trying to actually control how genes are expressed. So let's start at the beginning. We have Mendel. This is Gregor Johann Mendel. He was a monk. So he during that time, because around 1860s, most of the not exactly most, but centers of learning can actually be centered around the church. So Mendel's during his work in cultivating garden peas, he was working with plants, so genetics of plants. He was crossing different types of varieties of garden peas. He established this it's called regarded as a law, but three basic principles. You have the law of segregation, where every individual possesses two alleles and only one allele is passed to the offspring. We have the law of dominance. One allele will mask the effect of the other. One is dominant over the other. And the other is independent assortment, that the alleles of one character segregate independently of another character. During the time of Mendel, gene is an unknown term. He called those inheritable traits as basically a factor. But let's look at terminologies right now with regards to this. Speaking of these laws, we have experienced some terminology. So let's look at the terminologies first before we continue. So we have gene as the basic unit of inheritance. So it provides us an idea of the trait. So for example, a color of the petals, the skin, how tall or how short the plant is. So an organism carries two copies of the gene. That's one postulate of Mendel. Actually, it's in the law of segregation. You have two alleles and only one allele is segregated to the offspring. But an organism carries two copies of these genes. Now we have the allele. The allele is a variant of the trait. So the trait is the gene, so it's a variant of the gene. So for example, if the gene is about, or the trait or the gene is about the color of the petals, the allele is whether the petals should be white or purple. And then we call monohybrids as individuals having two different alleles of a single gene, or basically we usually have this thinking, or rather in doing genetical studies or genetic calculations, monohybrid crosses, we are tracking down a single allele or a single gene. Then we have your gametes. Gametes are specialized cells that carry the genes between generation and carries only one copy of the gene. That is tied to the first law, which is segregation. Only one allele is passed to the offspring. So how is it passed is through the gametes. The gametes carry only one copy of the gene, and it's the gamete that is passed to the offspring. And then we have the somatic cells. The somatic cells are the cells of the body, and it carries two copies of the genes. Now, let's move on to the second principle. So we have the law of segregation. So in the law of segregation, two alleles of each gene separate or segregate during gamete formation and then unite at random, one from each parent at fertilization. Why does it have to separate and then unite in the end? The reason is because it's in order to maintain the same number of chromosomes, the same number of genes, the same number of DNA or genetic information throughout the generations. Because if it does not segregate into this haploids or gametes, or just one copy, for example, when it combines during fertilization, it will become 4, and then a second round it becomes 8, and then another round it becomes 16. So it grows exponentially, and the cell cannot contain that much DNA. So of course there must be some what is passed through is just only one copy, and another copy is from another parent. And this is also a fact of sexual reproduction. In sexual reproduction, one copy comes, half of your DNA basically comes from the mother, half of the DNA comes from the father. It also provides you with varieties on the resulting offspring. And if you remember ecology, a greater variety provides higher chance of survival for the organism or even for the species as a whole. So basically, in this law of segregation, it actually supports principles of ecology. It also supports basically the idea of maintaining the same number of chromosomes or genetic material throughout the generations. So what is passed through is only a copy. Basically, half of your father or half of your mother is passed through to you. But your mother and father comes, the genes of your mother and father comes together to form yourself. So you have, it segregates during gamete formation. So originally, a person has two copies. During gamete formation, it segregates into two cells, each containing a single copy. And only one of this copy is passed through the offspring. The other copies comes from the mate or the another individual that it does sexual reproduction with. So that's the law of segregation. Now, the law of dominance. So remember, we have several alleles, or a gene is a trait, and we have alleles as the variations of the trait. Now, what if you have a copy of two different alleles? What happens then? So according to the law of dominance, if you have two unlike unit factors, which is the gene, responsible for a single character are present in a single individual, one unit factor is dominant to the other, which is said to be recessive. So basically, if you have two copies but different types of alleles, one allele is dominant and the other is recessive. And what is actually expressed is the dominant allele. And this comes another terminology, another set of terminologies. So you have the phenotype, which is the physical expression of the trait. So in here, you have here a heterozygous individual, which means an individual that contains two different alleles in his or her genotype, or where a genotype is the designation of the genetic makeup of the individual for the traits or traits it describes. So the genotype is basically it gives us the idea of all of our alleles. So you have the dominant yellow and the recessive green allele. So we usually write the recessive allele as a smaller letter, and the letter is designated by the starting letter of the dominant. So it's Y and not G, because the dominant is yellow, but it's small letter Y for green because it is the recessive one. So this heterozygous individual, meaning it contains a dominant allele and a recessive allele in his genotype, which is its genetic makeup. This is the genetic makeup. So what is its phenotype? The manifestation of the trait is it resembles or it favors the dominant one. So yellow is the dominant trait, so the phenotype is color yellow. So it shows as a color yellow even though it carries the gene or an allele for green. So that is the law of dominance. So the dominant trait shows rather than the recessive is being masked. So homozygous, by the way, is when both pairs of allele are the same. So here is the homozygous dominant, and here is a homozygous recessive. So for a recessive trait to show, that individual must be a homozygous, must have a homozygous genotype. So that is how your recessive gene would be expressed according to the law of dominance. And the test cross is a method to determine a known genotype by mating with an individual homozygous for the recessive allele. Because genotype determines the genes or the alleles for the gene that we have. And so because of the presence of recessive alleles, it does not necessarily mean that whatever you show on the outside, which is basically your phenotype, is the same as your genotype. So we must have this test cross method to determine if you have a mask, a genotype, or rather, if you have a recessive allele that is being masked, because it doesn't show in your physical form. Then the third one is the law of independent assortment. So during gamete formation, different pairs of alleles segregated independently of each other. So far we have talked about only a single trait, which is basically a color of the seed. Now we have, what if we have multiple traits? I mean, an organism has many different traits. So you have a yellow seed, but you are a tall plant with purple flowers. So those are three different traits. Now, according to the law of independent assortment, your alleles are sorted or segregate independently of the other. So basically, in this one, one trait does not have anything to do with the other one. Later studies will show that it is not always true, it is not always correct, but you have this interaction, or the general idea is that the gene is basically independent of other genes. So here, for example, are two traits, the Y and the R. So the Y trait and the R, the trait for the Y and the trait for the R. So when doing independent assortment, you basically get four sets of possible combinations, because of course the cells carry a pair of your, or rather a single set of your genes, but that doesn't necessarily mean that it only carry one gene. It carries one copy of all of the genes that you have. So that means if you have two traits, so two copies for each trait, you get four different combinations. So if it's three different traits, you will get 2 raised to 3, 8 different combinations. So that are possible allelic combinations according to the law of independent assortment. And these combinations, whatever those combinations might be, will be passed on to the offspring. So one way to apply these principles is to use the Punnett square, named after the one who proposed this, Reginald Punnett, which is a diagram to easily visualize the genotypes and phenotypes resulting from combining gametes during fertilization. So by convention, we have the male at the row at the top and the female on the horizontal columns on the left. So you have, for example, homozygous dominant yellow and a homozygous recessive green yeast. They form these gametes. So the first generation, so the P is the original generation. F1 is the um, first generation of offspring from the P generation. F2 is the second generation of offspring. So first offspring generation, so you have basically just a simple both of them are heterozygous. And then combining to this one, so because Punnett square is applied, especially in cases like if you have heterozygous individuals, so you can easily see the segregation. So the genotypes of the male on the top, the genotypes of the female on the other side, and then combine these two, you basically have 3 yellows and 1 green. And you have 1 homozygous dominant, 1 homozygous recessive, and 2 heterozygous offspring. So that doesn't mean that you always have this set of parents from the F1 generation always have 4 offspring. One is always green, the 3 are always yellow, but that just means it's a probability. So what this principle tells us, or this Punnett square diagram tells us, is the probability of producing such an offspring. So it's an application of the law of the probability. So you have a cross of this, for example, a cross of this yellow, green is the one that is apparently, no, it's not. It's yellow that is the dominant one, and the wrinkled is the recessive. Green wrinkled. Across of this. Yeah, the round, sorry. The round is the dominant. The yellow round is the dominant. So you cross these two generations, you get your different gamete formation. So basically, in the F1 generation, you will get a yellow round cross. So basically, this is just a simple application of a probability. So let's look at another one. This is a dihybrid cross for the green and yellow from before. So this is the green, 2 parents, heterozygous, both of them for both traits, for the yellow and round, but they are heterozygous. So what are the segregations? What are the possibility of their genotype? So you have 2 traits of 4 possibilities due to two independent assortment. So you have green wrinkled, containing both yellow and round alleles. You have yellow and wrinkled alleles. You have green and round alleles, and green and wrinkled alleles. And then the same goes for the other side. And then you combine this one. This is homozygous, both homozygous for the dominant one. This is yellow but heterozygous for the wrinkled. This is still yellow, heterozygous for the color but homozygous for the wrinkled. This is the offspring is heterozygous for both. So basically, you just combine all of them, you end up with this fraction, which is basically a probability of having this particular set of offspring. So this is basically application of our Mendel's law. So in the next part, we will look at other what is beyond the principles of Mendel.